Okay, so I think we'll go ahead and get started. It is now noon. Welcome, everybody. It's great to invite you back to the Connect Science seminar series. Again, thanking Claudia Steele O'Sullivan for all the work behind the scenes to keep the meeting going. A few housekeeping notes I'll make first is that please remember to post your questions into the Q&A tab, not the chat tab. And I will be able to get to those questions at the end of the seminar. And anything that we don't have time to address, Claudia will send those questions to Dr. Oliver to answer offline um, in that context. And please remember that the talks are also recorded, so you can check out the website that you see listed on your screen and check out any of the past talks that have been given in this great seminar series. So with that, it is my distinct honor and pleasure to introduce a great colleague and a friend, Dr. Trudy Oliver, who is coming to us from the Huntsman Cancer Institute. Trudy got her her PhD at Duke University and then took her talents to MIT, where she did her postdoctoral fellowship with Tyler Jacks and had a very successful postdoctoral fellowship to make great insights into lung cancer progression and development. And she's now she's an associate professor at the in the Department of Oncological Sciences at the University of Utah, and she's also a Huntsman Cancer Institute endowed chair in cancer research. She's also the co-leader of the Lung Cancer Disease Center at the Huntsman, and really, Dr. Oliver has had an amazing career already, very prolific in her publications, especially during a really challenging year of COVID. Her team has made some really seminal insights into lung cancer progression and development, taking a deeper dive into MYC and P53 signaling in that context, to not only understand disease progression, but also really understand and help define molecular subtypes of lung cancer that are clinically meaningful. And we're really excited to have Dr. Oliver here today to present her work to us. And with that, I will turn the screen over to Dr. Oliver.
Thank you, Sandy, so much. And thank you to the organizers for this awesome opportunity to present in this forum. I have to say, of all the negatives during the COVID period, this seminar series has really been a bright light. So I thank you all for connecting us through science in this way. And on to the title slide, I'm going to tell you today about how MYC drives small cell lung cancer subtype plasticity and hopefully show you that this is a not so small cell lung cancer problem. I have no financial disclosures. So on Twitter recently, someone said these introduction slides are boring, but I have to tell you that this is the most important slide to me. This is the reason why I get up in the morning. Small cell lung cancer is a neuroendocrine type of tumor, and in 2012, it was designated as a recalcitrant cancer by act of Congress because we've made so little progress in treating it over the last 40 years. These tumors tend to be very centrally arising. They're highly aggressive, highly proliferative, and metastatic from the beginning. And you can appreciate this from the image of a woman who came into HCI just about 2 years ago. This is the lung tumor burden in the chest area here, and this is a massive amount of liver and peritoneal metastases. These tumors like to go to the brain and the bone, lymph nodes, and liver, and you can appreciate they're incredibly PET avid and proliferative. And this woman also had a 2-centimeter brain lesion, as you see here. Now, we've been treating it the same way basically for the last 40 years, and that is with combination chemotherapy, as well as radiation and recently approved, though, immunotherapy. And this is usually a platinum compound such as cisplatin plus etoposide. And the only good news for these patients is that when many of them receive chemotherapy, you can see that there are dramatic responses to chemotherapy in about 60 to 80% of tumors. Unfortunately, we never fully eradicate the disease. The tumor cells come back, and at that point, they tend to be cross-resistant to most things. And we have, excuse me, we have very few second-line targeted therapies. So resistance is rapid. The average survival time now for these patients is about 12 months, and most people don't survive 2 years with this disease. To make matters worse, as you can appreciate here, surgical resection would be basically futile. So there aren't a lot of surgeries, and that means we haven't had a lot of tissue to study the disease over the years. As I alluded to, small cell has historically been treated as a single disease. There is no genetic testing or patient stratification in the clinic. And one of the main reasons for that is because these tumors really lack kinase alterations that are readily druggable. And instead, what we see is uniform loss of two key tumor suppressors, RB and P53. This occurs in virtually 100% of these tumors. And again, instead of kinases, what we see are alterations in transcription factors and chromatin regulators, which are difficult to drug. And in particular, we tend to see a gain of MYC family members, either through chromosomal amplification or overexpression. What I have listed here are the percentages of tumors that have chromosomal amplification. And so this is a small percentage of tumors, but we know that tumors can gain MYC through other means. So for example, c-MYC, which I'll refer to as MYC, is overexpressed in about 20% of small cell. The vast majority of the rest of the cases have high expression of its family member L-MYC, and then more rarely we see overexpression of N-MYC. And again, these tend to be mutually exclusive. Now, even though we treat it as one disease, back in the 80s, we had some of the first inklings that there is heterogeneity among small cell. And that is when the pioneers, Dr. Adi Gazdar and John Minna, were making some of the first cell lines from small cell. And they noticed when they took cells out of patients and put them in a dish that the cells would grow in two very different morphologies, either these tightly floating spheres like neurospheres, or in these more loosely attached, I call them bicycle chain-like patterns. And they named these variant small cell. And interestingly, when they took these cell lines and they injected them into nude mice, they maintained these differences in histopathology. So classic small cell looks like the typical, what we call small round blue cell tumor. Small cells, you can't really see into the nuclei. They're pretty uniform. Whereas with variant small cell, the cells were a little bigger, they have more open chromatin, and you can actually see prominent nucleoli. Now, over the next 20 years, a number of correlations were made with these cellular behaviors. So variant small cell was more commonly derived from patients who had relapsed on therapy or at autopsy. They were associated with shorter survival time. I told you it's a neuroendocrine tumor, but when they looked for neuroendocrine markers, the variant small cell had reduced expression, including ASCL1, which is a master driver of neuroendocrine fate. These cells proliferated faster. They tended to be more radio and chemo resistant. And you can see these are all hallmarks of really tumor aggressiveness associated with this fate. And importantly, this variant small cell was associated with c-MYC amplifications as opposed to no MYC or some other MYC family member. Now, fast forward to the 2000s, and some of the first mouse models of small cell were created by Anton Burns' lab. And not surprisingly, these models were based on loss of RB and P53 loss. And this led to tumors that look and behave like small cell, although the models took a long time to develop. And this prompted other investigators to get rid of additional tumor suppressor genes or to overexpress L-MYC or ASCL1. Importantly, all of these models develop what we call the classic form of small cell that look like this. And they have ASCL1, and so they're very neuroendocrine high. Now, to be honest, my lab was just prompted to work on this and to try to make a faster mouse model because we were struggling to do some of the key experiments we wanted to do with the long latency of these models. And so we decided to put in c-MYC into the RB-P53 mice. And when we did this at the time, we had a surprise, and that is that the mice did develop classic small cell, but they also developed a lot of this variant morphology. And we found these tumors to be ASCL1 low, or what I might refer to as neuroendocrine low or non-neuroendocrine. And around this time, serendipitously, we ended up talking to Martin Sauss in Germany, who was also studying small cell and paying attention to MYC status. He had done this small drug screen where he treated cells with various compounds and then noticed that the cells here in green, which have high c-MYC, responded exquisitely well to two drugs, alisertib and barasertib, which are Aurora A and B kinase inhibitors, respectively. And so with Martin, we decided to look in a larger drug screen that the NCI had put out with 63 human cell lines, and they had performed gene expression analysis so we could group their cells according to whether they were c-MYC high or c-MYC low. And you can see again with both alisertib and barasertib, the cells that had high c-MYC had a significantly lower IC50, suggesting that they're responding better to Aurora kinase inhibition. And I think the experiment that really nails this point down, also in collaboration with Martin Sauss, was that we took p53 RB null mouse tumor cells and used a CRISPR activation-based approach to either turn on c-MYC, n-MYC, or l-MYC. And you can see by gene expression, we can do that here, and we validated that at the protein level. But most importantly, only the cells that had high c-MYC expression responded exquisitely well to alisertib, as shown here. So really suggesting there's something about c-MYC that promotes this Aurora kinase vulnerability. Our lab decided to validate this in a preclinical or test this in a preclinical type of trial. And so we enrolled our RBP53-MYC or RPM mice into treatment with either PBS control, the standard of care combination chemotherapy, alisertib, or the combination of chemo and alisertib. And you can see in this micro CT imaging that we've pseudo-colored the tumors in this kind of yellow color. And I hope you can appreciate that over 12 days, this is an extremely aggressive model. These tumors go from barely detectable to a massive amount of tumor, and they are also metastatic. Now, by 20 days of treatment, you can see that in the first 3 treatment groups, the vast majority of animals have all significantly progressed on therapy. 1/4 of animals treated with chemo still have some kind of response, but pretty much all of the animals on alisertib and the untreated animals have progressed. But the combination of chemotherapy and alisertib, now we see about 3/4 of animals have stasis or regression at this time point. And that ultimately led to about a doubling in survival for animals that got alisertib in combination with chemo, as shown here. Now, around the time that we were publishing this data, the first clinical trial with alisertib in small cell was being presented by Tafika Wanikoko's group. And this was a trial, like many trials, in relapsed small cells. So these patients had already failed on chemotherapy. And there were 89 unselected patients that then went on to receive Taxol or Taxol plus alisertib. And these results look like many clinical trials in small cell, which is that there's no significant difference in overall survival. We're talking about 6.1 months compared to 5.4 months. But importantly, these investigators went back after the trial and looked at MYC expression by immunohistochemistry for the samples that they could get tissue for. And so they had 46 samples that they could classify then as either c-MYC positive or c-MYC negative. And now you see this complete inversion of the data, where the patients that had tumors that were c-MYC positive, that got alisertib, survived significantly longer than those who did not receive alisertib. This is about a doubling in survival from 2.2 months to 4.6 months. And you see the opposite in patients that had c-MYC negative tumors. So really showing consistently from cell lines to mouse models to clinical samples that MYC high small cell is preferentially sensitive to Aurora kinase inhibition. So to summarize this first part, we found that MYC in vivo can promote this neuroendocrine low, what we call variant type of small cell, that is ASCL1 low and appears to be very vulnerable to Aurora kinase inhibition. And at the time, this was really our first sort of a paradigm shift in how we were thinking about small cell, first originally as a single disease, and then we sort of began to realize that small cell appears to be more than one disease, or at least having different molecular subtypes, and that information would be important in the clinic. And so, you know, this is how we've been thinking about non-small cell lung cancer for a long time, where genetic testing is common and this information is used to apply targeted therapies to specific tumors. And really for us, this was the first time we started thinking about small cell as having molecular subsets, in this case, defined by mixed status. And so this really caused us to take a step back and say, you know, well, how else might these small cell subtypes be different that we didn't appreciate before? And if we look at them from unbiased perspectives, like their apoptotic profile or metabolic profile or immune profile, are they different? And of course, I don't have time to share all of the data that we know about these different functions today, but I will say the answer to all of these questions is yes, they are fundamentally different in all of these ways. And I'll give you a little vignette about what we know in terms of metabolism, because our lab is still actively working on that question. So this was also a really fun and productive collaboration with Ralph DeBardinis at UT Southwestern, who was thinking about small cell with some of the pioneers there. And his lab had been looking at human small cell lung cancer cell lines. And he noticed that the cells were metabolically distinct based on whether they had high or low expression of ASCL1. And as I've alluded to, that's also a very good surrogate for mixed status, because MYC and ASCL1 tend to be inversely correlated. So we decided to collaborate with Ralph and look at this in our mouse small cell tumors, because we had models that were either high for c-MYC or high for l-MYC. And again, you can appreciate that these tumors from a metabolite perspective are metabolically distinct. We can put those metabolites into a pathway analysis as we've done here. And we've followed up now on a number of these findings, but I just want to highlight that we noticed arginine, proline, and urea, which are intimately related from a functional standpoint. And so our lab decided to focus on this arginine observation. And to do this, we did a very simple experiment. We took our panel of human small cell lines that differ by mixed status, and we plated these cells. And then once they were happy, we took away the media and replaced them with either full media or media depleted of arginine, leucine, or glutamine. And then we measured proliferation a few days later. And this is both the raw data here by individual cell line, and then the data grouped according to mixed status. And what you can see when we take away arginine is that there are some cells that really crash and burn and die without arginine, and others that don't care as much. And when you group those cells by mixed status, you can see it's the cells that have high c-MYC that are really exquisitely sensitive to losing arginine. And this seems to be, you know, not just related to proliferation rate or any amino acid, because when we did this with leucine and glutamine, we did not see the same trend with c-MYC. And so we decided to test this concept. We wanted to test this concept in vivo, and there are a number of ways to deplete arginine in vivo. The two major ways are listed here. And this is through two enzymes that are pegylated. So in this case, this is a pegylated form of arginine deaminase, which converts arginine to citrulline and ammonia. Alternatively, you can use a human arginase 1, which causes arginine to be converted to ornithine and urea. Now, in data I don't have time to show you, we had also found that MYC-driven small cell was highly dependent on polyamines. And so we did not want to leave ornithine around, which is a precursor for polyamine biogenesis, for fear that the tumors would use that. And so we decided to go with this compound ADI-PEG20, which was provided to us by Polaris. And I'll say here that it was already well known, you know, arginine is the semi-essential amino acid. Most of your cells in the body can make their own arginine if they have expression of this enzyme ASS1. And it's known that tumors that lack expression of ASS1 become arginine auxotrophs and very dependent on arginine for survival. So we decided to test this compound in our classic GEM models. These are the mice that have high expression of LMIC. So in the RPR2 model, when we treated mice with ADI-PEG20, you can see there's absolutely no impact on overall survival. This also occurred when we treated a second model, the RPP mice with ADI-PEG20. You can once again see no difference from control animals. And this is in contrast to mice that got combination chemotherapy, which does significantly extend survival. But it was very interesting to us when we gave this compound to our MYC-driven RPM model. You can see now that ADI-PEG20 leads to a significant survival benefit compared to control animals. This actually performed better than the standard of care chemotherapy. However, when we combined ADI-PEG20 with chemotherapy, we did not get an extension in survival, so they didn't cooperate together. And part of our ongoing efforts now are to study the mechanisms of resistance to this drug and to use that information to create better rational combination therapies that do improve this response. And maybe not so surprising, the most obvious mechanism of this response seems to be the right one, and that is that in our MYC high models, as shown here, compared to two different LMIC driven models that lack c-MYC, we see that there's this complete loss or substantial reduction in expression of ASS1, which means that these tumors should be highly dependent on exogenous arginine, as they appear to be. We do know that when these tumors become resistant, they restore expression of ASS1, and we're studying those mechanisms now. Okay, so to kind of summarize this part, but leave you with why we still really care about this therapy, our lab has done a lot of preclinical drug studies in the RPM model. We've treated this very aggressive model with a lot of targeted therapies that have at some point been hot in the clinic. And you can see that the vast majority of them do absolutely nothing or very little incremental changes in survival. And I'll stop here on combination chemotherapy because this is the standard of care. It leads to about a doubling in survival in our mice. And it's really not until we create triplet drug combinations and combine chemotherapy with other targeted agents that we start to see these incremental shifts in overall survival. And then there's ADI-PEG20. So this is really substantially more effective in our model and why we're still excited about it and hoping to find rational combinations that make it even better that might be useful in the clinic. And so to kind of summarize this first part, once we realized that small cell differed by mixed status and that this seemed to be therapeutically relevant, we went on to look at tumors in a number of different unbiased ways. And a lot of that data is summarized here from our lab and the work of others. And I've highlighted some of the therapies that we found to be specifically effective in c-MYC high small cell. Recent groups work also suggests that this group may be the group of tumors that's most responsive to immunotherapy, which I think is particularly exciting. I will say that most of these findings are still really at the preclinical stage, but the aurora kinase example has been validated in the clinic, and we're working hard to try to move some of these findings to the clinic as well. And so around the time that we were having this revelation about small cell, really the whole field was starting to come to coalesce around the idea that small cell differs not just by mixed status, but by expression of some very important lineage-related transcription factors shown here. So I've already mentioned ASCL1. This is a master driver of neuroendocrine fate, and that's known. But from a molecular standpoint, the field had noticed that there are other key transcription factors that help stratify three smaller groups of small cell. And that is NeuroD1, which is a known driver of neuronal fate, PAL2F3, which had been shown recently to be a master driver of the tough cell fate, which is a different cell type in the mouse lung and human as well. And then a fourth subtype characterized by high expression of YAP1, although I will say some groups don't detect YAP1 by all measures. This group has also been called a EMT high or inflamed group of small cell. And importantly, these two groups, the PAL2F3 and YAP group, are very non-neuroendocrine, consistent with the fact that they have basically no ASCL1 expression, whereas these other two groups with ASCL1 and NeuroD tend to be more neuroendocrine-like. And as I've kind of alluded to, these three smaller groups of small cell are those that have high c-MYC expression, whereas the ASCL1 group tends to have high L-MYC expression and to lack c-MYC. So at this point, our lab became interested in this question of, well, what factors are determining these small cell molecular subtypes? And we came up with three possibilities that I will say are not mutually exclusive. One of those ideas was that these subtypes could be coming from unique cells of origin. And this partially made sense because, as I said before, ASCL1 is a master driver of neuroendocrine fate, and it marks neuroendocrine cells in the lung. And so perhaps these tumors are coming from neuroendocrine cells. Likewise, PAL2F3 is a master driver of tough cell fate and marks tough cells. So maybe those tumors are coming from tough cells, and maybe the NeuroD and the YAP subtypes are coming from different cells of origin. Alternatively, maybe all the cells are coming from the neuroendocrine cell, for example, but maybe it's the genetic or epigenetic program that the events that occur in the cell that determine whether these cells move down to a ASCL1, NeuroD, or YAP or PAL2F3 positive state. Alternatively, perhaps these cells actually are plastic, and that these states are related, and that one state may be able to convert to another with the right environment or genetic or epigenetic changes. And again, these are all possible and not mutually exclusive. So to get at this question, we decided to go back to the mouse model and just start with asking, what subtypes do the mouse models develop? And as I told you before, pretty much in all of the original mouse models that had been made, these tumors naturally acquire high expression of L-MYC. They'll actually commonly amplify L-MYC in these tumors, but all of them develop this classic morphology, are very neuroendocrine high, and tend to express ASCL1. We wanted to ask the question in our MYC high model, since these mice develop variant small cell and tend to be ASCL1 low, do these tumors express NeuroD, PAL2F3, or YAP, like is seen in the human disease? And I have to tell you more about how we do this in the mouse. So in the mouse, we initiate these genetic lesions by having mice inhale viruses or apply them intratracheally. And in this case, we have cell type specific promoters that drive Cre recombinase, and we use adenoviruses to carry these. And at our disposal, we have a number of cell type specific Cres. So for example, we have a CMV Cre that should theoretically be able to target any cell of origin, CGRP Cre, which is active in neuroendocrine cells, so the ASCL1 positive cells. And then cells that are more in the distal part of the lung, the club and alveolar type 2 cells, we can target with CCSP Cre or SPC Cre. And we decided to start with the neuroendocrine cell because that had previously been shown by Kate Sutherland's group that this is a predominant cell of origin in the classic small cell models. And so we're going to start with looking at CGRP Cre in either our MYC high RPM model or the next fastest model, the RPR2 mice that have high L-MYC expression. And so our question really is, using this neuroendocrine cell of origin, which subtypes of small cell do these different mouse models develop? And this is going to be a busy slide, but I'll walk you through the key points. So what we did is that we took our MYC high model or L-MYC high model, and we looked for expression of neuroendocrine markers, including that subtype marker ASCL1 and a number of other markers that tend to follow ASCL1 expression. And consistent with the fact that we targeted neuroendocrine cells, when tumors were just starting at the in situ stage in both models, you can see that they highly express ASCL1 and other neuroendocrine genes. However, when the tumors got bigger and more invasive, we see that in the MYC L high model, they maintain expression of neuroendocrine markers. and that's quantified over here, the third and fourth bars. But in the MYC high model, we see this dramatic reduction in neuroendocrine markers as the tumors progress, which you can see here. And then we saw something different when we looked at the non-neuroendocrine markers. And that is that in both models, when the tumors were small and in situ and just starting, they were negative for these non-neuroendocrine markers. But only in the MYC high model here do we see a gain in these non-neuroendocrine markers, including the subtype marker NeuroD and YAP, markers of mesenchymal fate like ZEB1, and then markers of Notch and REST signaling, which has previously been shown to promote non-neuroendocrine fate. And we'll talk more about that later. But this didn't happen in the RPR2 model, the MYC low model. They never expressed these non-neuroendocrine markers as quantified here. So that left us with two questions. One, you may notice that I didn't mention that fourth subtype marker, PAL2F3. That's because it was not detected in the tumors initiated from neuroendocrine cells in either model. And then secondly, this prompted the question of, well, are these ASCL1 positive tumors in the MYC model actually converting to the NeuroD and the YAP subtypes in the presence of MYC? So we wanted to address both of those questions, but to just summarize what we see so far, just like in the human disease, the LMYC associated models develop the ASCL1 positive group of small cell, and in the presence of CMYC, with the neuroendocrine promoter, we see expression of NeuroD and YAP, but we did not detect PAL2F3 positive tumors. So we wanted to know, what is going on with the PAL2F3 subtype, and do we perhaps need to hit a different cell of origin? So we went back to that capacity to target different cells of origin with different promoters driving CRE, and we repeated these experiments in both models. And what I'm showing here is in the MYC-L high model, it didn't matter whether we used a neuroendocrine promoter or that CMV promoter that could target any cell, we never detected PAL2F3 positive tumors. However, in the MYC high model, not when we use the neuroendocrine club or AT2 promoter, but when we use the CMV promoter, we did detect some PAL2F3 positive tumors. This was about 7% of all tumors. And this sort of leaves, while we don't know what cell of origin we're hitting here, it leaves open the possibility that we're actually hitting tough cells and that those may be the cell of origin for the PAL2F3 positive tumors in this model. So now to summarize, really, I think the mouse models do a great job recapitulating the patterns of small cell subtype expression that we see in the human disease. And we can now say that PAL2F3 positive tumors can be driven by MYC, but in some unknown cell of origin. And so that left us back to our original question, does MYC actually have the capacity to drive small cell from an ASCL1 positive state to a NeuroD and or YAP positive state? And to address that question, we had to come up with a different assay. And Abby in the lab decided to take our RPM mice at early stages of tumorigenesis. This is before you can detect overt tumors by micro-CT. In fact, all you see at this stage are in the large airways, you have these little polyps or little in situ tumors as magnified here. And you can appreciate when we take serial sections that these tumors are ASCL1 positive at this stage. They don't have NeuroD or YAP. And then Abby dissociated the lungs from mice at this stage, got them into a single cell suspension, and then cultured them for different time points in a dish. And when we did this, we saw something pretty interesting. And that is that when you do this experiment with a MYC-L high associated model, you put the cells in the dish, they grow as these tightly floating spheres, and that never really changes no matter how long you grow the cells. However, every time we did this with cells from the MYC model, you can see that they start out in these tightly growing spheres, but then over the course of about 20 days, they convert to this bicycle chain variant looking morphology. And you can really appreciate that this nicely recapitulates what is seen in the human cell lines that I showed you earlier. So Abby decided to collect these cells as they were transitioning in the dish, or presumably transitioning in the dish, and to look at them for our favorite neuroendocrine and non-neuroendocrine markers over time. And you can see that at these early time points, these cells express ASCL1 and other neuroendocrine markers. They also express NeuroD. And around the time the cells are downregulating, or we're seeing the absence of expression of neuroendocrine markers, we now start to see a gain of non-neuroendocrine markers, including YAP, as well as markers of the Notch rest pathway, which I told you before is a known driver of non-neuroendocrine fate. And this is not just an artifact of mouse cells in a dish, because when Abby took human classic small cell lines and overexpressed a stable form of MYC, we see that this frequently leads to downregulation of ASCL1 and induction of non-neuroendocrine markers like YAP and Notch signaling. These particular cells did not turn on NeuroD, but we have other cell lines that did. And another group, the Watanabe group, recently published very similar findings in a different panel of human cell lines. So really suggesting that MYC can drive cells away from the ASCL1 state to other subtypes of small cell. So to get at this with finer resolution, we decided to perform the same experiment and capture these cells at 6 distinct time points for single cell RNA-seq. And you can see that we analyzed over 15,000 cells, and they're grouped here according to day. So day 4 to 7 cells are here, day 11 cells are here, and day 14 to 21 cells are located here. And the nice thing about single cell is that we can perform these pseudotime analyses, which will relate these cells and their sort of transcriptional relationships. And importantly, this approach predicted a single sort of evolutionary trajectory from early to late pseudotime that satisfyingly agreed with the time point data. So at day 4, most of cells are in early pseudotime. By day 7, they start to round the corner here and start to go into late pseudotime. By day 11, many more cells are in late pseudotime. And by day 14, they're pretty much completely out of early pseudotime and in late pseudotime. And that is consistent at day 17 and 21. Now, if you look at ASCL1, NeuroD, and YAP, our subtype markers in pseudotime, you can again appreciate that ASCL1 and NeuroD are expressed early, and YAP1 comes on around the time those factors are disappearing. So we next wanted to know, again, is this just something that happens uniquely in a dish, or is this sort of transcriptional evolution happening in vivo? And to address that, we decided to isolate 4 individual tumors from the mice and also perform single cell RNA-seq. And these are presumably clonal populations, because we're taking out single individual tumors and performing single cell. And you can see that in all 4 of these tumors, they showed the presence of cells throughout that entire trajectory that we saw in vitro, with different percentages of cells in late pseudotime in a given tumor. But what this really suggested to us is that these individual tumors appear to have subtype heterogeneity within a given tumor. And to further validate that, Abby looked at a quantitatively bazillion number of serial sections, where you can see we did this at the in situ stage and the invasive stage. And basically in these serial sections that are, again, presumably clonal, we see that in situ stage, at the in situ stage, tumors mainly have ASCL1 and not NeuroD and YAP, but by the time the tumors get bigger and more invasive, we start to see expression of all of these subtype markers. And that's really quantified here. So at the C2 stage, we predominantly see one subtype, and that subtype is ASCL1 positive. A small number also have some ASCL1 and NeuroD. But in the bigger, more invasive tumors, now we tend to see 2 or more subtypes. That tends to be ASCL1 and NeuroD in green, or all 3 markers in orange. So this suggests as well that small cell subtype is impacted by tumor subtype plasticity, and that MYC has the capacity to promote cells away from the ASCL1 fate to the NeuroD and YAP states. So our next questions that we wanted to answer is really, how is MYC promoting this subtype plasticity, and does this occur in human tumors? And so to take the first question, we decided to look at our single cell data in a deeper way. And a published neuroendocrine score had been published by Adi Gazdar's group, and we applied that neuroendocrine score to our cells in the single cell RNA-seq analysis. And you can see down here, these are our day 4 and 7 cells. They're very neuroendocrine high, and those were placed in early pseudotime. And you can see that over the course of pseudotime, they lose their neuroendocrine fate. That's easily quantified here in the violin plot. And you can see there's this pretty dramatic shift in neuroendocrine fate from day 7 to 11 in our cells. And so we really wanted to focus on what is going on in this, this big step in neuroendocrine fate between day 7 and 11. So we took that data and looked for transcriptional regulators of that particular switch. And at the top of the list, we saw REST, which is a known transcriptional repressor of neuroendocrine fate. And it was very familiar to us because, again, my colleague Julian Sage had nicely shown that the Notch REST pathway can promote non-neuroendocrine fate. In fact, the Notch pathway is frequently lost in small cell because it is a known regulator of ASCL1. And so loss of function of Notch presumably activates ASCL1 and keeps cells in a neuroendocrine fate. And so we saw REST as coming up as a potential regulator of this fate switch, and that made sense to us. Moreover, if we looked at known Notch inhibitory factors in our single cell data, you can see that these Notch inhibitory factors are expressed in early pseudotime very strongly. And then around the time these inhibitory factors go away, we started to see expression of pro-Notch signaling genes like Notch2, HES1, and REST itself. Moreover, we performed ChIP-seq for MYC, and we found that MYC can actually directly bind some of these pro-Notch signaling genes like Notch2 and HES1, as shown here. And they're actually conserved EBOX sequence motifs consistent with MYC's ability to bind these genes and turn them on. So suggesting that MYC is contributing to small cell evolution by turning on Notch signaling. And we wanted to test this functionally, so I'm going to show you the pharmacological data here. We treated both cells and mice with 2 different gamma secretase inhibitors, DAPT and DBZ. I'm showing the DAPT data here. So recall that normally these RPM cells in culture go from the classic to the variant state. We see that when we apply DAPT, that this helps keep the cells in the classic state in these floating spheres much longer. And consistently, if you harvest these cells and look for our neuroendocrine and non-neuroendocrine genes, you can see that we maintain the neuroendocrine fate markers at a higher level and longer. And in contrast, normally non-neuroendocrine genes emerge over time. And when we treat with DAPT, we either block or seriously delay expression of these non-neuroendocrine markers. We see something similar in vivo. So when we block Notch in vivo with the same drugs, we see a morphology that resembles the classic stage of small cell more, smaller cells. They also express higher levels of ASCL1, consistent with them being blocked in their evolution. And moreover, this significantly inhibits tumor development. So by micro-CT and histology, we see significantly reduced tumor burden when we treat with DAPT over the course of this experiment. And as I told you, at the genomic level, Notch frequently has loss of function mutations in about 25% of tumors. You can appreciate as I zoom in here that these are mutually exclusive alterations in Notch 1, 2, and 3. And so we decided to look at whether this made sense, whether our hypothesis made sense in the human data. And so what I've done here is grouped Notch mutations by either wild type or mutant status. And then I've also labeled whether tumors are MYC high or MYC low. And then we've applied our neuroendocrine score to each of these tumors as well. And I think the first thing you can appreciate is the difference here, and that is that the Notch mutant tumors are uniformly neuroendocrine high. And that's consistent with losing Notch, keeping ASCL1 on, and keeping cells in a neuroendocrine state. However, when tumors are Notch wild type, they can be both neuroendocrine high or non-neuroendocrine. And you can appreciate that in the ones that are non-neuroendocrine, the majority of them have high expression of c-MYC. consistent with our hypothesis that c-MYC can take advantage of wild-type Notch to push them into this non-neuroendocrine state. And from a contingency table perspective, this is a highly significant finding. Again, consistent with our idea that MYC requires Notch to drive small cell evolution. Okay, so to kind of summarize our view of the state of the subtypes, we see that this group of these three smaller groups of small cell that have high c-MYC expression also tend to be those that are Notch wild type, whereas those that have high L-MYC are more likely to have Notch mutations. And so finally, we wanted to know whether this plasticity that we see in the mice also occurs in humans. And so we addressed that in a few different, hopefully complementary ways. One, we took advantage of the fact that there's a lot of bulk RNA-seq data out there for human small cell tissue and cell lines. And so we applied cybersort analyses, which allows us to basically ask within an individual tumor, what percentage of the tumor resembles our MYC-driven stages of subtype evolution. And the important thing that I want you to see here is just that it predicts that the bulk of tumors actually have a lot of transcriptional heterogeneity with presumably cells that represent different states within a given tumor. And satisfyingly, for example, in the YAP1 group, we see the highest amount of the latest stages of MYC-driven progression, as we would predict. Secondly, we were able to acquire, at the time, one human small cell biopsy. And of course, these presumably arise from a single cell. This was provided by our collaborator at WashU in St. Louis. And you can see in this single sample analyzed by single cell RNA-seq that there are two distinct populations of cells, one that has high ASCL1 and one that has high NeuroD within the same tumor. And satisfyingly, the ones that have high NeuroD also have high c-MYC, consistent with the data we've shown here in mice. And then finally, we looked at this at the protein level by IHC in a small subset of human tissue that we could acquire HCI. So we had 21 human biopsies and we looked at their status of MYC, ASCL1, NeuroD, and YAP. And that data is just summarized here. So we do see some tumors that are only ASCL1 positive or only NeuroD positive, but we also see a nice handful of tumors that have two or more subtypes within a given tumor that again are presumably arising from a single cell. So suggesting that there is subtype heterogeneity. This study was too small to really correlate with MYC status, but I will say we did have the trend of the samples that had high c-MYC were more likely to have two subtypes, whereas those that were MYC low were more likely to have one subtype. And I think this has been largely confirmed in a much larger study from Sloan Kettering just in recent days with 174 samples. So to summarize what I have hopefully shown you is that using mouse models and single cell approaches, we find that MYC can drive small cell from this ASCL1 state to this NeuroD state and eventually a YAP positive subtype. And we think at least one of the ways MYC does this is by directly turning on Notch signaling, which is a known inducer of the non-neuroendocrine fate. And then I think we've also seen that a number of factors come into play in determining small cell molecular subtypes. One is cell of origin. So remember that we only found PAL2F3 positive tumors when we targeted an unknown, potentially tough cell of origin, whereas we could get these other subtypes from a neuroendocrine cell. Genetic program also matters because we could only develop NeuroD, YAP, and PAL2F3 positive tumors when we used c-MYC as opposed to L-MYC. And then finally, we see that cell fate plasticity appears to occur because MYC can drive cells from the ASCL1 state to the NeuroD and YAP positive state. And I think this has some really important clinical implications because it really suggests that small cell is a moving therapeutic target. And I think now the way we view things is that we would really like to be able to control this transcriptional landscape. So for example, can we create barriers that keep the cell into a particular state and not able to move around so much? Secondly, we know that we're often encountering tumors that have cells that are in different states already. And so, and we know that these states have different therapeutic vulnerabilities. So how can we find a way to perhaps funnel these cells into a more uniform state so that we're statistically more likely to make a difference when we apply a given therapy? And in regard to therapy, I just want to say one last piece of data that we had sort of found before we appreciated all these things. And that is that in chemotherapy treated tissue, in both human samples and in human cell lines, upon chemotherapy resistance, we see this trend of reduced ASCL1 protein expression, reduced ASCL1 message, and reduced expression of ASCL1 target genes, suggesting that during chemotherapy, cells are moving into different small cell states. And so with that, I feel like we've kind of come full circle from viewing small cell originally as one disease to realizing, oh, there are these distinct molecular subtypes and this information really matters for therapy, to coming back around to this concept that, yes, that's true, but also this seems to be one very plastic disease. And so I think we have a big challenge ahead in tackling this problem, but I think we have a better understanding now of how it works. With that, I want to really thank the extremely talented, very hardworking graduate students who contribute to all of this work today. Gurkan Malaglu, who started the mouse model work and who's now with Miriam Murad. Milan Chalishazar, who started all of the metabolism work and who is now at Merck. And that work has been taken over now by Gabby Fort in the lab. And then I spent a lot of time today talking about Abby Ireland's work, who published our recent cancer cell paper and really did the lion's share of that work. I want to thank our entire lab, really a number of fantastic collaborators and awesome funding agencies for supporting our work. I thank you for your attention, and I'm happy to take any questions.
Great, thank you, Dr. Oliver. That was fantastic. I only wish you could hear the applause and the feedback for what was a phenomenal talk. We have a few questions, and thank you. You've left sufficient time for us to have some good discussion. So let me get to the questions. Chiho Kim is asking, since ASCL1, NeuroD, and PAL2FE are transcription factors, MYC could change the levels of transcription target genes of them in parallel with their expression change, ?
Yes, that's absolutely true. I mean, we see that the transcription factor levels themselves change during MYC-driven progression, but MYC really binds a lot of things. We see that by our ChIP-seq analysis, and so it's probably contributing to expression of different genes as the cells sort of epigenetically rewire during this process as well. So yes, I think all of those things are probably true, if I understand.
Yeah, I think you did. Thank you. Candace Lay is asking, how did you build the signature matrix for CyberSort?
Oh, that's a great question for Abby Ireland, who knows how to work the computer at a far greater level than I do. Just from the very basic level, we divided we had a lot of bulk RNA-seq data and single cell data of the different stages of MYC-driven evolution. And so we grouped some days together according to their behavior, and that created a signature for each stage. And then we applied that data to the bulk RNA-seq data from the Nature et al. Paper. But Abby if you contact us, Abby can definitely give you the more nuts and bolts answer to that.
Great. It sounds like a really useful tool. So Rafid Nakash, and please pardon me if I'm mispronouncing some names here. Phenomenal talk. Question, IO plus gamma secretase inhibitors in YAP1 high, ? Assuming.
Say that one again.
Yeah, so I think the question is about the use of IO plus gamma secretase inhibitors in YAP1 high tumors.
Oh, okay, yeah. So immunotherapy plus Notch inhibitor. So theoretically, so I'll say one, there's a practical challenge with Notch inhibitors, and that is that they're very toxic. They're hard on the GI tract, and we can only give them for so long. And I'm imagining that that's probably a problem in people as well. So we're kind of limited in long-term experiments with those drugs in mice. There's probably some next generation things we could explore that do a better job. But the point is that Notch inhibitors help keep the cells more in the ASCL1 positive state. The new data in the field suggests that it's the later stages that might benefit from immunotherapy. So that combination may not work so well, but I think you're on the right track in that we need to be thinking about how we can control or contain these cells in different phases of evolution, and then hit them with the things that they're vulnerable to in that state. So I think from a concept perspective, that's exactly what we're thinking about now. I don't have any great answers today, but stay tuned. I hope that's what we devote our next 5 to 10 years to.
Yeah, great. Candice Lay is asking again, how does the plasticity transition state in establishing biomarkers in determining which therapy to use?
Yeah, you've hit on another concept that we are thinking a lot about, which is, can we take advantage of this information about subtype to make a difference in the clinic? There's a lot of efforts going. So as I alluded to, we don't get a lot of biopsies from small cell. It hasn't been sort of the standard of care. Biopsies do carry a little bit of danger to them in a sense. And certainly surgeries are extremely rare. But one thing I will say is that because these patients have highly metastatic disease, we can easily take a blood sample, and that is, cheaper and faster and safer, and we can do it throughout the course of therapy. And so there are a lot of efforts going into how can we use blood-based biomarkers, and that could be circulating tumor cells, circulating free DNA, whatever you imagine. People are trying to use that and see whether we can find information about subtype or hopefully mixed status from the blood, and then to, for example, enroll patients, for example, MYC high patients, specifically to the targeted therapies that we've identified. And our group at Huntsman is really motivated to try to do that for arginine deprivation therapy, because it has been tested in the clinic, but it was a very small trial. They were not pre-selected. We don't have any information about mixed status, and really it was too small of a trial to know what to expect in terms of whether there were truly any MYC high patients in that group or not. So we really want to kind of do that trial in the right way and test, for example, whether arginine deprivation would be effective in the MYC high patients.
Mm-hmm. Yeah, a lot of work to do. And maybe I can ask my own question here, because you kind of touched on it. A lot of times in other solid tumors, we're able to do gene expression profiling or single cell RNA-seq on tumor at different stages and normal adjacent tissue. And I wonder if you have the luxury of even being able to do that, or if you have, perhaps in your mouse model, has that given you any insights about the relationship? I think a lot of that might relate to the microenvironment, or the stroma is where you might get the most information there. But is there any information you have on how those early lesions compare to normal tissue?
Yeah,
to get at a cell of origin or development issue.
Small cell grows so fast that I think, the vast majority of patients come in and they already have metastatic disease. So I think when we do catch small cell tumors that are actually early, it's usually some kind of like accident in the clinic that people were looking for some other reason and they found it, because, as I sort of showed in the mice and in people, it's really just so metastatic from the beginning. I think it's like part of the nature of these cells that they're migratory very early on. So I think it's hard to study the earliest stages in human. But The tumors do tend to be very centrally arising, which is where neuroendocrine cells are located in the central part of the lung. So I think all of the data together makes sense that the neuroendocrine cell is probably a predominant cell of origin. After all the plasticity I've seen in mice, I'm not convinced it's the only cell of origin, but I think it's probably one of the most frequent and common, sort of easy to be cells of origin.
Yeah. And perhaps slightly unrelated, but do you have a sense, is the microenvironment vastly different between the subtypes?
We don't know much about that. I feel like that's not been very well explored yet in the field, but with the advent of single cell, I think there's a lot of power in that technology that, now when we're analyzing tumors in our different mouse model and we do single cell, we just go ahead and get all of, take the whole tumor and get a lot of interesting information about cells that are in the microenvironment and certainly the immune cells. And I think that's a really important area for further study in small cell because it's actually thought to be a very immune cold tumor as a whole. Not a lot of immune infiltration, not a lot of antigen presentation. And so there's only this very small subset of patients that are currently responding to immunotherapy. That's in the smaller group that we believe are MYC high. It's been seen in what they're calling the, inflamed subset of small cell, which I think overlaps a little bit with YAP and PAL2F3 and MYC. So I think one question going forward is, how can we try to stimulate a warmer immune microenvironment for the vast majority of tumors that don't have that? So I think it will be fun to take advantage of these new technologies to better understand both the immune microenvironment and maybe the role of other factors there.
Yeah, great. We have a couple more questions that we'll get to, and then we'll have to do a hard stop in a few minutes. But Jagpreet Nanda says, fantastic talk. Can you reverse the YAP or other end phenotypes by removing MYC somehow in already developed stages, or is the MYC effect permanent?
Yeah, that's a great question. We really are interested in how reversible are all of these different changes. And we haven't done enough in that regard. So we have positions open for those who want to come think about those questions with us. I can't say that we've done those studies. The bulk of the data in the field suggests that it's not that hard to get cells to go from neuroendocrine to non-neuroendocrine. I've seen fewer examples of non-neuroendocrine going back to the neuroendocrine state. I have heard, I think, an example or two in a cell line where that seems to happen, but we certainly don't see it spontaneously under any conditions in our model. But we haven't done a lot of the experiments that you're suggesting, for example, getting rid of MYC. I'm pretty sure Abby did the experiment where we inhibited Notch after the cells already have high Notch and have converted to non-neuroendocrine, and that is not enough to make them go back. I can say that. So, yep. But these are all really important questions.
Yeah. Maybe one final question, because it may relate to that idea. From Rafi Nakash, any role of epigenetic modulation using HDAC inhibitors in MYC high? There's Encore 601 data of Pembro plus etinostat in MYC high non-small cell lung carcinoma in exploratory analysis.
Interesting. Yeah, I think this is a really important future direction too. I can't speak too much about it because we just don't know the answers at this point, but undoubtedly there's some serious epigenetic remodeling going on as cells convert from these very different states and using very different powerful lineage specific drivers. So we know that there's a lot of profound epigenetic changes. I think it's a really important question of whether we can capitalize on some of these pharmacological tools that modulate epigenetic state to help control this and maybe make some therapies more effective. So great area for future study.
Great. Well, unfortunately, we've run out of time. We do have some more questions in the Q&A, and Claudia will send those to you to look at and discuss offline if you wish. So once again, I'd like to thank you, Dr. Oliver, for joining us, making the time to present your phenomenal work in a really challenging area. And best of luck to you going forward. Thank you.
Thank you, Sandy, and thank you everyone for your time today. Bye-bye.